All right, so for this audio lecture, we are going to be looking at cellular structures that are found in both prokaryotes and eukaryotes and comparing those. This is not a comprehensive overview of all cellular structures that are found in prokaryotes and eukaryotes. Again, the focuses are of a handful, fewer than a handful, structures that are found in both and doing a comparison of the two. And the first structure we're going to be looking at is the ribosome. So keep in mind the ribosome, its function is to allow for protein synthesis to occur. So you are going to have message RNA or RNA binding to the ribosome. And then the ribosome will end up taking the information that's in the RNA, reading in codons in groups of 3 to determine which amino acid to select to then bind to the previous amino acid to then create a polypeptide chain to then eventually a protein. The protein would then be folded so that it could then function. Okay, but the ribosome itself has proteins in it and also RNA. So you will see the reference to ribosomal RNA, or little r, and then big R, big N, big A. So that is ribosomal RNA, and that is looking at the RNA that is part of the ribosome. Generally, it is the small subunit that is looked at, or the SSU. So if you see SSU, little r, RNA, that is talking about the small subunit. And depending upon the organism, that fragment might be different. So for prokaryotes, that small subunit, the ribosomal RNA, is 16S. So if you see 16S, you are looking at a prokaryotic organism. If you are seeing 18S, that indicates that you're looking at a eukaryotic organism. Okay. In our image here, you can see how we have in the top figure, that is a prokaryotic ribosome. So again, you can see how there is the small subunit. Now, when we consider the ribosomal RNA and the protein, the size of the small subunit in a prokaryote is 30S. And then there's the large subunit, which is kind of in that middle part. And so that is 50S. It has protein and RNA. And when these two complex together, so when the 30S and the 50S complex together, it ends up being 70S. So when proteins complex together, they tend to actually get a little bit smaller because they're binding to each other really tightly. And that's why it's not an 80S ribosome, it's a 70S ribosome for prokaryotes. Now compare that to a eukaryotic ribosome, which you see in the bottom figure. You can see how there is the light green and then the blue indicating the two subunits. And so for a eukaryote, the small subunit with protein and RNA is 40S. And then the large subunit with protein and RNA is 60S. When those two come together, it is not 10, 100x S, it is 80S. So again, when you have these two complexing together, they've just become a little bit smaller in size. And so, again, really important here is that prokaryotic ribosome is going to be 70S. A eukaryotic ribosome is going to be 80S, right? So the size of those subunits are different. The sequence of the RNA is different. So again, we can, as mentioned in the endosymbiotic audio lecture and also in class, if we were to sequence that ribosomal RNA, that small subunit of the organism, we can actually create these like phylogenetic trees and group them and determine when common ancestors happened and then when there was a divergence from that, because that sequence is unique for organisms. If they, we can also use that to determine, is this the same species? Now, the function's the same for both prokaryote and eukaryote. It's the site of protein synthesis. Now, when we look at genetic information, we said that all cells have genetic information. How that genetic information is packaged is a little different. So again, if we look at a prokaryotic first, they have a single chromosome. It's circular. It's double-stranded DNA. So that chromosome is located in the cytosol, and it's just kind of aggregated together, and it does get clumped or packaged around proteins, but they're different from histones, and it's not as organized. So it's kind of like if you took an elastic band, which is circular, and then you kind of like clump it together and make it so it stays really small. That's kind of what's happening in a prokaryote. That single chromosome has all of the critical genes that cell needs in order to survive. So there's not a lot of like luxury type genes, accessory genes. It is like, what do we need in order to be biochemically active, undergo metabolism, and survive on any given day. Okay. Now, bacteria can take up plasmids. Plasmids are extra chromosomal DNA, so it's not included in that circular piece. They are small, separate areas. They usually have a gene or two, and these are a little bit more like luxury genes. It makes the bacteria's life a little easier, a little nicer. So it might code for antibiotic resistance. On every single day, the bacteria is not going to be coming up against an antibiotic, but if the host ends up taking antibiotics, really nice to have that plasmid that codes for antibiotic resistance. You could turn that, you could express that enzyme and then deactivate that. So as mentioned in class, I likened the bacterial chromosome, that large circular piece, as like the four wheels on your car, right? A car needs to have four wheels in order to function. It needs an engine, right? So there's critical things that a car needs in order to get you from point A to point B every single day. Now, there's certain things that are really nice to have. So like plasmids would be like the heated seats, the heated steering wheel, or the cooling seats, the spare tire, right? Most, on the average day, you probably don't need those, but it does make life a little nicer having those. Now, when we compare that to eukaryotic cells are going to take its genetic information and enclose that in a membrane, the nucleus, right? That membrane has nuclear pores, and if you look at that figure, that nuclear pore is not just a hole. There's actually this network of proteins that act like a basket, so it can control what enters and exits into the nucleus. So genetic information, the DNA is going to stay in. The DNA, the chromosomes get wrapped around histones and other proteins, so it is packaged and organized very neatly, if you will. And that's really important because most eukaryotic organisms are going to have multiple chromosomes, and they're going to be linear. And so, that elastic band that we had the analogy for the bacteria, if you put a whole bunch of elastic bands together, it's kind of easy to separate them. If you were to take pieces of string and just put them into like a bowl or something, they're going to end up getting intertwined and knotted and everything. And that's why the packaging of these linear chromosomes is really important, so that they don't end up getting like knotted with another chromosome where there might be a breakage, and breaking of the chromosomes could lead to new organisms, which could be beneficial. But at the same time, if we want an organism to maintain itself as it copies its genetic information and such, and utilizes the genes, again, we want to make sure that is protected and packaged. So again, you can see how there's similarities between the genetic information, there's differences. And obviously genetic information is what's utilized to make RNA to then make protein. All right, the other structure we're going to look at is the flagella. So we'll talk first about bacterial flagella, and then we will compare it to eukaryotic flagella. So bacteria flagella, and really both flagellas are going to allow for motility. So the movement of the cell that it's attached to from one location to another and towards a stimuli or away from a stimuli. Okay. Flagella for bacteria, it can lead to virulence factors because if you have a bacteria that can get from one location to another location effectively, that can allow you to be more virulent or cause infection more easily. The pattern of bacterial flagella is unique to the organism. So we can have it where there's one flagella, which is monoturcus, turcus meaning hair, so one hair. We can have polar flagella, so flagella at the end of the cell. So if we have something like a bacillus, it would be found at one end. We get an amphiturcus, so think of amphibian, both land and water, and amphiturcus would have hair on each end of the cell. So again, if we had a bacillus at both ends of that bacillus bacteria, there would be flagella present. Lophoturcus, lopho meaning clump, and so there's a clump of flagella, a cluster at one end or both ends, but there's going to be multiple flagellas in that area. And then periturcus, peri throughout, so periturcus, there's hairs throughout, so they're going to cover the entire surface. What does this look like? So in this figure from the textbook, you can see how our first bacteria, which is actually H. pylori, which is found in the gut, it has lophoturcus. So you can see those flagella towards what is being shown is kind of like the bottom end of the bacteria. So as those move, that would propel that cell forward. In the middle panel, we have Pseudomonas, so that's monoturcus. So there's just a single hair that's coming off of it. That hair is a flagella. And then our last one, that is periturcus, and so you can see how there are flagella throughout the surface of them. So the structure of bacterial flagella versus a eukaryotic flagella are different. So in the upper figure, you're looking at a bacterial flagella. So it actually is made up of a single filament. It says one filament. And there's a basal body at the base of it. And that basal body acts as a motor, which allows for the filament to then rotate. So the movement of a bacterial flagella is going to be in like a propeller fashion in a circle. Okay. And again, there's one filament for each flagella. So if you had something where it was periturcus, you'd have these basal bodies with filaments coming out of it throughout the surface. But one basal body for each filament. And again, that propeller motion that you end up getting is unique to a bacterial flagella. Comparing that to the eukaryotic flagella, which you see in the lower figure, it actually has 20 filaments. So you'll notice that there's like a red and an orange. So those actually is 2 filaments. And there's 9 of them. So if you look towards the outside and count, there'll be 9 of them. So that's known as 9 times 2. And then in the middle, there's 2 additional filaments. That gets our last 2. So 9 times 2 is 18 plus 2, 20. So again, 20 filaments. It is going to have a basal body at the base of it. Keep in mind, it is organizing 20 filaments, not one. And there's going to be mitochondria near their basal body because the basal body needs ATP. And so you generally, with a eukaryotic flagella, you see a higher concentration of mitochondria towards that, near that basal body. Just to kind of backtrack with the mitochondrial flagella, remember that ATP is made on the plasma membrane. So across that plasma membrane is where ATP is generated. So you would end up having ATP right near that basal body. So that's why you don't need mitochondria. You will also notice on the eukaryotic flagella that there is a motor protein. So dynin is a motor protein, and so that is actually going to allow for the kind of the attachment and then pulling of the neighboring microfilament pair. And that ends up causing, as this is happening, it kind of causes like almost like a shortening and a lengthening on opposite sides. And it causes that structure, the flagella with these 20 microfilaments, to end up moving back and forth. So kind of in a waving pattern. Now, I'm going to go out on a limb, but most people probably have seen sperm underneath the microscope, and so that is probably the most common eukaryotic cell that has a flagella that most people have seen underneath the microscope. And so if you've seen either a live imaging of sperm or animation or something, that flagella actually has that kind of whipping back and forth motion. So it's not a circular motion like we see with bacterial flagella, it's a back and forth. The same would be true for like eukaryotic flagella that's like on protozoas, protistas, that we see that have flagella. Now, for a number of reasons, the movement of a eukaryotic flagella, a cell that has a eukaryotic flagella versus a prokaryotic flagella is different, right? So again, that waving back and forth versus the propeller motion. So when in this slide is talking about a prokaryotic flagella, because it's rotating like propeller, you're going to get what's known as a forward run or a run. And so it's going to move all coordinated in a forward, driving that cell in a forward direction. What's going to happen is that randomly that propeller is going to kind of not work as well. So maybe it's like, a boat and the engine doesn't have enough gas and the propeller's kind of like spinning along and the boat's not going to be propelled forward. Also, if we're talking about a bacteria that's got multiple flagella, like we see in panel D, there's going to be times where the flagella are not propelling in the same direction. It's not going to be coordinated together. So remember, prokaryotes, they don't have like a central nervous system, so they don't have a way to coordinate that movement. So when that flagella starts to propel in the opposite direction, when you have the multiple flagellas not working together, what ends up happening is the cell tumbles. So if you were to watch this underneath the microscope, you would notice in like live imaging that the cell would have this very forward motion, and then all of a sudden it would just kind of tumble around. It would have a forward motion and then tumble around. So again, this is referred to as this tumble, run, tumble movement. It looks pretty uncoordinated underneath the microscope, but it still will move towards or away from a stimuli, depending upon whether the cell needs to move towards it, or if it's a toxin, it wants to move away from it, right? And that process is known as chemotaxis. With eukaryotic flagella, the movement is more forward, and that is because of the waving motion. And you have motor proteins in there controlling that waving motion, so you don't tend to get a disorganized piece to it. One thing I'd like to add about the flagella for both prokaryotes and eukaryotes, they are made up of proteins. And so those proteins have to be synthesized, and then the flagella has to be assembled. So we do want to keep in mind, and I guess a little bit more with prokaryotes, that the presence or absence of flagella is not always an indication of what it will have at all times. When we're looking at them under the microscope, whether it's live imaging, whether it is a slide that has been prepared and the cells have been fixed, it is a snapshot of what's going on. So protein expression is dynamic. It is in response to stimuli, cell signaling events. And so depending upon the environment that the bacteria is in, and in some cases eukaryotic organisms, it may not be getting the signals to produce a flagella, so it may look like it has no flagella. But if there becomes competition, if there is a reason for motility, expression of flagellin, which is the protein that makes up flagella, can be turned on. It can be expressed. It can be assembled. And then we would have a cell that has motility and can get out of that situation or go into that situation, depending upon what type of bacteria it is and what that stimuli is. So if you do have any questions about any of these cellular structures, please let me know. Again, this is not every cellular structure. This is just meant to be some, to give you an example of some of the cellular structures that are found in both prokaryotes and eukaryotes, and then a comparison of them.